Ladies and gentlemen, first I would like to say thank you for the organizers importing us here. It's really an honor. And I'm sure that you are as excited as I am about our upcoming visit to the JGI, but first let me take your time to tell the story about Raman microspectroscopy and active single-cell sorting. This is a quite old picture, but I find it still astonishing because it really shows you the diversity and the close interactions, how microorganisms share space and might interact like neighbors and partners. And this in situ community structure analysis was, of course, a big topic only in the 90s, but already by that time, they were sure that the big challenge will be to assess the function of the microorganisms. Of course, metagenomics, metaproteomics, metametabolomics are really cool and important advances, but they are still insufficient to really get the function of certain microorganisms. Of course, we have a bunch of methods, though, to access the functional traits of yet uncultured organisms. We can label them easily with isotopes, and then go and identify them with fish, for example, and then detect this isotopic label with different methods like micro-autoradiography, nanoscale, secondary ion mass spectrometry, or Raman spectroscopy. Our department has a long history in using and even developing these methods, but let's say we needed something more elaborate, which is less time-consuming and is really compatible with subsequent analysis methods like genomics, and is free of cross-labeling. So we thought that the goal will be achieved with Raman microspectroscopy, which is really a non-destructive, high-resolution, and very sensitive method, which is also very fast. And you can really combine it with fish on the spot, because as you see, this instrument is basically a microscope, which is an epifluorescent microscope as well, and coupled to a spectrophotometer. So if you take a fish image, identify cells, and go to spots, you can really measure Raman spectrum from single cells. And this can be coupled to an optical tweezer, which allows you to manipulate cells in fluids. So just a brief introduction how Raman microspectroscopy works. If you bombard your sample with a laser beam, there will be photons hitting the molecules and the molecular bonds of your sample. And they will bounce back either elastically or inelastically. If they bounce back inelastically, they will give some energy to the molecular bond and raise it to a higher vibrational state or vibrational mode. So the bounced back photon will lose some of its energy, so the wavelengths will be shifted. So in a Raman spectrum, we focus only on these red-shifted photons. We call it Stokes scattering. What does a Raman spectrum tell to you? Of course, the wavelength of the backscattered photon will really refer to the vibrational modes and the bonds where they have interacted with. And from this, you can really assign different peaks to certain molecular bonds and certain molecules than in a Raman spectrum. But of course, a more complex molecule will be assigned to multiple peaks, like nucleic acids and proteins, for example. But certain complex ring structures will have only one really sharp peak, like the phenyl ring of the phenylalanine amino acid, or, for example, cytochromes, that you can really easily detect in a spectrum. If you have a heavier isotope in a molecule, of course, these bond energies will be different. So in a Raman spectrum, you will be pretty easily identifying isotopic labels. In this proof of principle study, we used 13C labeled phenylalanine on an organism which can incorporate this directly into its proteins. So we had a really sharp shift due to this label. But of course, you can utilize other compounds which have 13C or deuterium labeling them, and what the organism can degrade and build into its macromolecules, and then you will have other shifts in other peaks. But these labeled substrates are a bit expensive, so we went on to test the possibility to apply deuterium oxide, so heavy water, to label cells. And this isotope is really nice because you can use it as a general activity marker. Every microorganism uses water to its metabolism. And the most prominent label in a Raman spectrum of a cell is the lipid-carbon-hydrogen bond. And if the cell incorporates deuterium into its lipid, this bond will shift a lot into a normally flat region where it shows up as a carbon-deuterium bond, which is super easy to detect. And this incorporation rate is linear with the percentage of heavy water you used in your medium. And an additional beauty of deuterium label is that it is incorporated with a higher efficiency to autotrophic organisms, like these archaea down here, who are usually slow growers, so are hard to label isotopically. We recently published a study based on this labeling technique with some applications already leading to the single-cell regime, what I will shortly show now. The proof of principle we reached with focusing on complex carbon source utilization in mouse gut microbial communities. So we just took a cecum sample, put it with 50% deuterium containing media, and amend it with different carbon sources and incubate it for a short time. After this, we have measured single cells from each amendment and also performed FISH on these samples, targeting two different organisms, Bacteroidetes acidifaciens and Akkermansia mucinifila. In these box plots, you can see the individual labeling percentage of the single cells. Above this red line, we think a cell is labeled. So you can really see that these two organisms had a totally different pattern of activity in the different amendments. And it was also different from the bulk randomly measured cells. And yeah, the nice thing is Akkermansia mucinifila was most active on mucin amendment, which is no big wonder, but it really shows the power of this approach. Of course, we wanted to go a step further to isolating these active cells. And this we did in a capillary. We focused on mucin and glucosamine and used an optical tweezer to pick the cells with a nice Raman spectrum, which were labeled, and move it to the end of the capillary where we can cut them down and then use them for subsequent analysis, like whole genome amplification, and then, for example, a 16S PCR and clone library. And it worked out nicely, so we have found many organisms in this, let's say, very small metagenome, and also Akkermansia showed up mostly in the mucin, Akkermansia showed up mostly in the mucin amendments, while we have found a bunch of other organisms that we didn't expect to show up in these amendments as active. Can we do this in vivo as well? Yes, we can. So if you just feed a mouse with D2O, you can easily, within a very short time, find that the cells are labeled. So deuterium is quite easily applied to either living organisms or microcosms or enrichments or maybe even separated environmental samples. But throughput was an issue so far because measuring cells one by one with Raman took really a long time on the scale of 1 minute per cell and finding another cell. And also isolating these cells in a capillary took half an hour to an hour. So we combined our forces with an expert in the microfluidics field to really move to a high throughput regime. And this expert was Roman Stocker from the MIT. And the idea of this project was to develop a device that can sort out cells from various sample types. So you just simply incubate your sample with either 13 C-labeled substrate or an unlabeled substrate and add D2O to it, and then just pump it through a microfluidic device, which can measure cells in a very quick manner, so in 1 second, let's say, and which is cheap, so you can just throw it away after one sample to avoid cross-contamination. And this device should be really compatible with subsequent analysis and might serve as an input for the JGI single-cell pipeline. So this fruitful cooperation led to this beauty where you can simply put your sample into a syringe, pump it through the device, and the device will sort out cells on certain Raman properties into a tube where they can be cooled or put into a preservative, store it, and then sent to JGI for single-cell analysis. If we take a closer look into the heart of this machine, you can see that cells are coming in the middle of this channel and going to the waste branch. But in this region, we can capture them with an optical tweezer, move them out from the stream, measure them, and if they are labeled, we can release them here, where they will move to the sorted branch. If we don't like the cell, we can just put them back to the middle stream, and they will go to the waste. Of course, this really has a power if you can automate it. So Kang-So Lee and David Barry wrote a MATLAB script and an automation which can control the Raman instrument. And this works out quite nicely now, but we are beginning the accuracy testing of the system and, of course, to optimize it to various cell sizes. Of course, parallel to this high-throughput automated sorting development project, we try to push the manual sorting to its limit in a real scientific question, which I will talk about in the next part of my talk. Our group is mainly working with nitrifying organisms, both archaea and bacteria, which can drive the oxidation of ammonia into nitrite and then nitrate. This field is moving forward very quickly, and there is still a lot of explore. For example, the nice microdiversity, what you always see in wastewater treatment plants. Here you see 2 clone libraries of, or not clone libraries, but sequence trees of Nitrospira lineage 1 and 2, which are containing samples from only one single wastewater treatment plant, but from different years. So there is a huge stable microdiversity in this plant, and it really exists through a long time. How can it be so stable? Because this game is only oxidizing nitrate into nitrate, but cells can still coexist on this certain one process. So there must be a lot of small niche differentiation and maybe special physiological traits that keep all the players in the game. And if you look at this picture, which shows you only Nitrospira living in the close proximity of each other, you can really imagine that they should do something else to really stay in the game on this single process. We have already shown that they can be mixotrophic and even aerobically hydrogenotrophic and utilize different compounds like cyanate, for example. But it's still a black box, so there is a lot of things to explore. Why microcolonies? Because basically these microcolonies are, let's say, clonal clamps of microorganisms of a single strain. So if we isolate one microcolony, we'll have a higher chance to have a nice genome coverage of this certain strain, because it's a number of cells, let's say a natural amplification of the genome. But of course, if you singularize a microcolony, you will still have associated taxa, like NOBs and AOBs together, and even some other bacteria, which we do not know the function yet. And moreover, if you just sonicate a microcolony, a sample, and you find still microcolonies intact, you will find even predators attached still to the microcolony. So pretty exciting things going on a single microcolony. What we did is we incubated wastewater treatment plant samples freshly for a very short time with ammonia, and also added either 13C labeled bicarbonate or deuterium oxide to the sample. After incubation, we sonificated them, and then in the already mentioned method, we just put them into a capillary and sorted for the active colonies, do whole genome amplification and send it either to the JGI or just PCR check for nitrifiers. What traits did we use to look for nitrifying organisms? Luckily, all nitrifiers, AOBs and NOBs, both have a huge cytochrome content. So the cytochrome peaks, these black ones, will pop up really sharply in a Raman spectrum. And of course, as an activity measure, we used the C13 incorporation or the deuterium incorporation. First, we tried to use both of these methods, but then we settled with deuterium as it was really easy to utilize. Where did we get so far? First, after the first trials of these, let's say, development on 2 microcolonies, we really gained some confidence in the method. So we did do a training set of 24 microcolonies that we have already sent to the JGI, and 20 of them assembled really nicely and are already available for analysis. And then we went on by performing 2 different incubations with 2 different ammonia concentrations and isolated 140 microcolonies, which we are about to send here to the JGI. And these great bioinformatic guys did a bunch of analysis on this data set, which is already existing, and I will show some of it in the next time. And they also struggled a lot because these microcolonies cannot be treated neither as a single-cell genome nor as a big metagenome. So the software pipelines needed quite some tweaking to get it really going. What have we seen? If you look at these focus plots of the individual microcolonies, we can see that we have captured nitrifiers in all of the microcolonies nearly. And we have captured a bunch of other diversity which are closely associated to these nitrifiers. Basically, this Niabella showed up everywhere, so there must be some strong linkage between nitrifying organisms and them. But of course, one might ask, why is nitrifier coverage so low sometimes, and why we do not even see them in some of the microcolonies on these plots? Well, the answer might lie in the lysing efficiency, which is known to be very low for nitrate and ammonia oxidizing bacteria. So we can still improve in that. Did we find the microdiversity? The answer is yes. If you look at this tree of concatenated marker genes from our genome beans, we can see that we have found our target organisms, we have found diversity in them, and we have found 2 other big group of associated organisms, namely Bacteriates and these microgenomates. And we clearly have no idea yet how they are interacting, but we will look deeper into the genomes, but that's still future. And if you look at the nucleotide identity distribution for these various genome beans plotted against these reference genomes, we see that we have caught organisms which are not the same species as these reference genomes. So everywhere we have found something new, which is really exciting for us. Now, of course, the next step is to look deeper into these genomes and compare them to each other to find common traits and, of course, specific traits for example, the different ammonia concentrations where we have incubated and other differences. And what else exciting things did we find so far? In one of the ammonia oxidizer genomes, we have found a whole cluster of genes forming a complete type 6 secretion system. And this is the first ever discovery of a type 6 secretion system in an AOB. So What is it used for? It's usually used for interactions like invasion to a community or defense or signaling molecules and even to kill other microbes. So it's a bit of an exciting insight into the games going on within a microcolony. And I'm sure that we will explore much more novelties. And as an outlook, let me show you one other device that we are playing with now. We have already shown that the deuterium incorporation into a cell is pretty quick. So within a few tens of minutes, you can already have a detectable label. You don't even have to wait for the cell to divide. So if you design a microfluidic chip where you have your sample flowing in its original medium with the cells inside, and you can just grab out a cell, wash it in pure medium, and then incubate it with deuterated medium with a specific compound of interest in it, or maybe the normal compound and some antibiotics. And from time to time, you can just bring this cell here and measure it in pure water for its Raman spectrum to see whether it was able to use this compound, whether it's labeled. And of course, you can combine this with sorting. So immediately, you can release this cell here, where it will travel to a tube, and you can just preserve it immediately for transcriptomics. So we wanted to go real time with these activity measurements. And with this, I would finish my talk. And I would like to say thank you for the funding agency, especially the JGI and this Isotope program and the community sequencing program. And of course, to all our colleagues at the Department of Microbiology Ecology at the University of Vienna, and all of you for your attention. And I'm happy to take questions.
Any questions? So will the goal be to get things into 3D4-well plates? Is that the
Sorry?
Will the end product be a 3D4-well plate full of single cells? Is that what the...
The end product from us will be a bunch of cells from the original Raman sorter, what we can send here and can be singularized by your pipeline. So that's something that we would do here.
Bill?
This question may be a little bit too specific, and you may not know the answer straight off the top of your head, but in the mouse gut, you had cells that you were feeding something that were related to Barnesialaceae. Do you happen to recall what they were eating?
Sorry, I cannot answer this question here because I was not performing this stuff.
Yeah, sorry, I thought it might be a bit specific.
Yeah. It was, sorry, a bit too specific for me.
Okay, well, thank you very much. And this concludes this session.